Welcome to today's NFDI talk. This episode is about fair data in immunology. Today's speaker, Christian Busse, is a team leader at the German Cancer Research Center and speaker of NFDI for Immuno. He studied medicine at the University of Freiburg and did his PhD at the Max Planck Institute for Immunobiology. His work focuses on developing scalable and sustainable solutions for immunological research data management. Welcome, Christian. The floor is yours.
Yeah, thanks, Natalie, for the kind introduction, and welcome everyone also from my side. So, as already introduced, the title of my talk today is Fair Data in Immunology. And so what I would like to talk about over the next roughly 30 minutes is the following. So first, give you a quick idea on what our consortium, NFDI for Immuno, is aiming to do, as this will be helpful to understand also basically the remaining parts of the talk. Then we will have a quick look at the various types of immunological research data, then pick out two examples to see how fairness is already achieved or aimed to achieve for two of those data classes. Then I will share some thoughts about how to generalize data models, and then finally spend some time on challenges and potential solutions for the diverse types of immunological data that we're trying to deal with. So in general, I hope that everyone understands that 30 minutes, of course, is not a lot of time. So we will probably have to stick a little bit more to the surface that we would like to. But I hope that following talks of people from our consortium over the next year can then drill down a little bit more on individual aspects of that. So first of all, who is NFDI for Immuno? We are a consortium of the third round, so meaning that we successfully applied in 2021 and started like the other third rounds consortia in March last year. We have 10 co-applicant institutions and 5 participants. And when we built up NFDI for Immuno something like 4 years ago, one of our key goals was really to have a sufficient number of partners so that we would be able to represent all the different subdomains within immunology. And so therefore, what we really now can very well cover are things like cancer immunology, infection immunology, autoimmunity, transformation processes in lymphoid cells, veterinary immunology, and also computational immunology. In addition, our participants, so as I said, these are 5 of them, they kind of complement the skills and knowledges that we already have on the side of the applicants by bringing in additional expertise, like for example, allergy or bacterial pathogens. Okay, so and basically with this diverse set of partners, we think that we can really understand and represent the immunological community at large when it comes to their needs for research data management. So the aims, which are probably similar to those of most other NFDI consortia, is of course, first of all, that we want to make sure that immunological data is fair. And to this end, we're aiming to provide high quality annotated data sets that follow community standards, or at least try to, as we will talk about later on. All of this will be hosted on an interoperable infrastructure. And in addition, of course, we will also try to provide tools and training to enable the community to really utilize these research data management resources for their own research. One of the first questions that we needed to solve when we started was, of course, what is actually the scope? So what are all the data types that we need to cover because they are really uniquely important or of unique importance to people in immunology? And so to this end, we conducted a survey. This happened basically in the second half of 2021. This survey was relatively widely circulated, including an advertisement via the mailing list of the German Society for Immunology. And we managed to get a total of 180 or a little bit more than 180 completed questionnaires. So this is just the demographics to show you that we really have had relatively broad coverage across self-assigned fields of studies from the participants, and also widely ranging across career stages. So really from PhD student to department heads. Now, the key question that we then ask people is, what are the data classes and the data types that you're typically handling? And so the distinction that we're making here is that the data class is the biological property that is measured, for example, a cell phenotype. On the other hand, the data type would be or is determined by the experimental setup or the device that you measure, that you use to measure that property. For example, a flow cytometer. So cell phenotype as data class would traditionally be measured by a flow cytometer as data type. But of course, the flow cytometer can also be used to determine other data classes, like, for example, if you think about a Luminex assay, where you would then use the cytometer to determine cytokine levels. Or on the other hand, the data class of a cell phenotype can, of course, also be measured by other technologies, like, for example, microscopy. So this is basically the biological versus technical distinction, which is important to make if you think about metadata annotation. So when we ask that question, what are the data classes that you use basically daily, often, sometimes, rare, or never? We got the result that the following 6 data classes are the ones that are frequently used, so meaning that at least 50% of the respondents use them at least occasionally. And so first and foremost is really cell phenotype, which then also was determined that this is basically all of that is flow cytometry. Then in addition, protein and gene expression are a little bit lower with a wide variety of techniques. Antibody and TCR reactivity, which includes common assays like an ELISA, of course, also are encountered frequently. Spatial information like microscopy, and finally, adaptive immune repertoire, immune receptor repertoires, so short AirSAC, is basically the last of the 6, but still used by nearly 60% at least sometimes. The other interesting information that we got out of this is that the majority of respondents is really working in the human and the mouse system at the same time point. That was important for us to know with regard to the question whether we could rely on other infrastructures that are already providing services for, but focused on human data. And so this one clearly showed us that we need a place for mouse data because it's still very frequently generated, in addition to a smaller but still significant number of respondents who said that they're working with livestock like cows, pigs, or chicken. Okay, so with this one, the final list of data classes that we assembled was, of course, first the cell phenotypes, then the adaptive immune receptor repertoires, levels of soluble mediators like cytokines, chemokines, antibody and TCR reactivities, immune receptor ligands, So this is basically the counterpart to the previous point. So peptides within the MHC complex. And that is complemented by physiological observations and clinical data on the subject. In addition to this, we are aiming to have metadata overlays, and I will come to that, what that exactly means, to gene expression data and spatial information. The reason for this distinction is that especially gene expression data is something where there's already a lot of infrastructure for, both in terms of infrastructure that provides protected access, like the things that the colleagues from GHGH are building up, but also generic resources like Array Express, for example, or Geo, where such data can be already now deposited. Spatial information, as long as we're talking about photon-based technologies, then of course it's the realm of NFDI for bioimage, with whom we are closely collaborating, so that given that there is already some kind of infrastructure for this kind of data, what we would like to do here is basically keep or maintain the information that is relevant for an immunological interpretation of such experiments, which occasionally cannot be stored in the generic infrastructures. Okay, so now let's pick out basically the top two of the previous ones that we had here, so the cell phenotypes and the AirSec data. And let's have a look at how fair this data already is at the current point. And I assume that looking at the participants list, you don't need an explanation of what fair is. So findable, accessible, interoperable, reusable. So these core principles that we're all trying to achieve to basically as a general goal in research data management. So if you're looking at cell phenotype, then the community, which is mainly represented by ISAC, the International Society for the Advancement of Flow Cytometry, already passed the minimal metadata standard over 10 years ago, which is called MyFlowCyte, and contains several dozen items that are considered to be really necessary to interpret a cytometry experiment. So this, of course, minimal metadata standards are key if you're thinking about findability, because this is the key data that would be harvested. So we already have a check here. In addition, there is a community-operated data repository, which is actually run by ISAC. That's called Flow Repository, which unfortunately has already collected a little bit of dust. So ISAC is currently in the process of finding or building a replacement for that one. But it is currently still unclear what the technical infrastructure for this will be, as well as it is not yet clear who will finally operate this repository. But there is something that where this data in general can go, and this, of course, is important if you're thinking about accessibility. So the A and FAIR. Right now, there is no common API to request data from these repositories. You have a user interface, but in terms of programmatic access, which is, of course, one of the key components of the FAIR principles, this is currently lacking for cell phenotypic data. Data representation, the thing commonly known as format, is clearly one of the success stories for cell phenotype data, because the FCS, so the flow cytometry standard format, it really has been around for decades and is, in contrast to what we know from other basically technological domains is universally used by equipment manufacturers. So the compatibility or the interoperability at this point is relatively high. There is no data schema that really would tie the information both from the minimal metadata standard and from the data representations into some kind of higher order structure that would be helpful to understand the information semantically at this point. And then finally, derived metadata. This is the term that we use in NFDI for Immuno to describe so-called top-level analysis results. So when you run a typical analysis, then there are a couple of things that most people would look for that would drop out of such an analysis. And while it is a downstream result, it is metadata in that regard that most people would like to search for it when they're looking for data sets. So therefore, this is something that is usually very helpful for users to identify the things that they're looking for, and therefore we're calling it metadata. To simply to flag that fact. Okay, if we compare this with basically with what AirSec can provide, there we have the AIR community as a small but international society or group of people who has been working on standardization basically since 2015, 2016. We have minimal metadata standard, which is called MyAir, which was published end of 2017. There is a set of data repositories, which is called the Air Data Commons. This is a federated network of repositories, which are basically held together by the common API, which is the so-called Air Data Commons, ADC API. This API looks like a REST API. If you're very deeply into REST APIs and you look at the definition, then you will see that while it does implement, of course, a get and a post function, it does not necessarily behave completely like a REST API, but which has been criticized by some people. But in general, it does the job of helping you to identify data sets and query data sets from the repositories very well. And it has the special feature that it also allows you to query within single individual data sets so that you can, for example, only download a given subset or a given fragment of the data that is present in one data set, which can be helpful if data sets are large. There is a data representation standard, which is called AirTSV, that contains all the add-on information that is usually necessary to interpret this data with basically fixed column names. And again, this is a tabular format. The whole thing is, over the last 3 to 4 years, has been embedded into a larger definition, which is the Air Data Schema, that then also contains additional concepts that are helpful if you're trying to interpret this data, like, for example, clonal structure, like receptors or even individual cells. So that therefore the whole thing is relatively flexible if you want to integrate it with other data sets. And then finally, derived metadata can be represented via or is already included in the AirTSV, and so therefore can already be queried for in the data repositories, which typically ingest this AirTSV. Okay, and sorry, what I forgot previously. So the missing derived metadata, or the reason why there is a question mark for the flow cytometry here, is because while there is a standard called CLR to represent a population frequency, which is the thing that most people would like to see here, the standard is basically has been published 11 years ago, but has hardly ever been used. So that therefore, while it is there, it's not used, and this is the reason why it's labeled in orange here. Okay, so now these are two examples on how basically already fairness criteria are tried to, or basically how well things can be structured in immunology. The truth is, of course, that for a lot of other data sets that we talked about, especially things like receptor reactivities, cytokine levels, and so on, unfortunately, there is hardly any community standard at this point. And so this brings us to the question, how can we, if we have all those multiple community standards, how can we bring those standards together in a meaningful way? How can we interconnect them? And the way how how we look at this is that basically for all community standards, typically try to define the data of their subject really top to bottom. So basically from what is called a study here, really to the bottom of whatever their individual technology provides. And now, if you look at those lower columns, which are called AirSec, cytometry, ELISA, and some random future technology here, then there will be a set of information or metadata that is completely specific for a given measurement technology. If you think about cytometry, where you have individual channels that are detected by PMT settings, where you have fluorochromes, where you have antibodies to use for staining, all this information will not be available for an AirSec experiment simply because it doesn't exist there. So meaning that there is a boundary between an upper and a lower layer, and the lower layer basically is this, then consists out of these columns, which are technology specific, and therefore we call it sample data and metadata layer, because of course the actual data, measurement data is also contained here. But we also have what we call the biological metadata layer. And this can be usually structured into these three categories, what we call study, subject, and sample. So study would be the most top-level information, who did it, what's the title, a couple of keywords. This is basically also what would be, for example, in DataCite or Dublin Core, roughly. Then information on the subject, additional information on the sample, and then basically the sample is then used for a given measurement. Okay, so now importantly, semantically, there is a very good matching between the individual standards and how they define it, so that we think that it is possible to harmonize between the existing community standards. And when harmonizing, then of course you can also extend this, basically the layer that you have here, onto measurement technologies that right now do not yet have any community standard. Okay, so with this one, I would then basically switch gears final time or last time, and then talk about potential challenges that already we might have already identified. So first thing, of course, is how do we handle data classes and data types without standards? This is basically what I already mentioned, that we can try to start with the biological metadata layer, because the information there can still be applied to such data, and then basically try to work downwards from there, which means identifying the information that is then really missing to make these things useful. The second question is, of course, the interoperability between the existing standards. I already said, okay, we can map and try to harmonize. But of course, ultimately, we need to put in a lot of efforts to work with the communities who originally defined those standards to make them aware that their standard doesn't exist in a vacuum and that they need to take up the basically the task to see with which other standards they basically can get closer with, so that the interoperability in general improves, and that the loss when doing the conversion between two standards gets lower and lower. The one thing that is important to stress here, that the one solution that does not exist, although some, I think everyone would like to have it this way, is to have one large overarching standard, because this is typically, while of course it would be capable of solving a lot of the issues that we have with these individual community standards, the adoption of such large standards is basically, with all the experience that people have made over the last years, very, very low. And this is basically the critical component that you have based on the community standards, that they are very focused on one task, and therefore they're perceived as relatively useful by people in the community. So we really need to try to work with those standards, not trying to replace them. Question number 3, and this is of course something that is really keeping us awake at night if we're thinking about operating real repository infrastructure, is what is actually the granularity of data access that a repository needs to provide. So I said before that, for example, the Air Data Commons has a very special feature of allowing to select individual lines from a data set, or basically from one sample, and this is a very nice feature. On the other hand, looking at the usage data, you also see that it's very rarely used in practice, because people still tend to download the complete data set once they have identified it. So people might be interested in searching for features, for internal features of a data set, but then still would download the data set as a whole, which potentially also has something to do with, because you want to have it, you want to have non-matching entries as an internal control. So the reason why this is important is that if you consider to be the sample level access, so meaning that you can access individual files that represent the measurement from one sample, if you consider this to be the sweet spot in terms of granularity, so being able to go fine enough into an individual within a complete study, but on the other hand, not complicating things too much, because you still have this file level access and your repository doesn't really need to know the internal structure of your data, then this really gives you the possibility to use generic repository software stacks like DSpace or like Immunology to basically provide access to such data. Second to last point, identity and access management. As you've seen in our initial survey, while 60% of respondents are working with human data, and while not all data might really completely fall under GDPR, because of course there's some types of information that you might legitimately be able to completely anonymize. We assume that there will always be some kind of information that would be considered personal data. So therefore, you clearly need to have access management here. And just to remind you that this does not run contrary to the FAIR principles, and especially not against the access principle, because there basically it says that you should be as open as possible, but as closed as necessary. So if you need to protect data, or if you cannot make data globally available due to privacy regulations, then this is just a good reason to do so. So for this one, we're, as a consortium, we are right now running a beta test of the IAM for NFDI structure. So this is basically the first base for NFDI service that is now entered the integration phase. And that basically provides a set of community AAI services that will then be all integrated via a proxy server. And so ultimately what this service will fully allow the NFDI to do is to have, for a community manager, to have one single interface where he or she can assign group memberships that then allow access to data across a set of federated repositories, so that the individual repositories don't have to do that detailed maintenance in terms of access management anymore. Final point, search portal integration. So this is, of course, the F in FAIR. This is one of the questions, again, on which level you want to have a search portal. Clearly, search portals for an immunological audience would provide much deeper access to information, so allowing queries much further down in the study subject sample stack that I mentioned initially. But one of the things that we really would like to focus on for wider discovery is integration with search engines like OpenAIR or others that use the OAI PMH and DataCite XML interface, so that studies in the repositories can be discovered widely by a frequently used engine. Okay, so with this one, I hope that I could give you some overview on where, from our perspective, things currently stand in immunological data management. Thank you for listening. Thanks to everyone who's part of the NFDI for Immuno team. This is a picture from our recent all-hands meeting 2 months ago in Berlin. And if you need more information, you can follow us via the NFDI website or basically on our Mastodon channel. Thanks a lot.
Thank you very much for the talk, Christian. Yeah, with that, we say goodbye to all the people on YouTube. In our next episode of the NFDI talk on 15 July, Björn Hagemeyer will talk about Jupyter for NFDI. So, bye.